Hola, bueno, vamos a empezar. Gracias por estar aquí en este seminario del programa de Frontiers en Genomics, que organiza la licenciatura en Ciencias Genómicas, el Centro de Ciencias Genómicas y el Instituto de Biotecnología de la UNAM. Y como siempre, las preguntas son al final. Los que están presentes aquí pueden alzar la mano, los otros lo pueden hacer en el chat y las preguntas en el chat van a aparecer en la pantalla, entonces todo el mundo las va a poder ver. Y bueno, yo les daré el micrófono para que puedan preguntar a los que están acá. Bueno, se los darán. Bueno, vamos a iniciar. Para mí es un gusto presentar a la doctora Katrin Karbstein de Scripps Biomedical Research en la Universidad de Florida. La doctora Karbstein hizo su licenciatura en química en la Universidad del Ruhr en Bochum, Alemania, en 1995. En 1998 obtuvo el diploma en bioquímica en la Universidad de Witten-Herdecke en Alemania y en el 2003 el doctorado en bioquímica en la Universidad de Stanford. Del 2003 al 2006 hizo un postdoctorado en la Universidad de Berkeley, en California, en el Departamento de Biología Molecular y Celular, bajo la dirección de la doctora Jennifer Doudna. Del 2006 al 2010 fue profesor asistente en el Departamento de Química y Química Biológica en la Universidad de Michigan Ann Arbor. Del 2010 al 2001 fue profesora asociada del Departamento de Biología del Cáncer en el Scripps Research Institute y del 2013 al 2017 también en el Research Scripps Institute con tenure track. Del 2017 al 2019 fue profesora asociada en el Departamento de Biología Integrativa, Estructural y Computacional en el Scripps Research Institute y desde 2020 a la fecha profesora del Departamento de Biología Integrativa, Estructural y Computacional también en el Scripps Research Institute en Florida. La doctora Karbstein ha recibido varias distinciones importantes y por tiempo solo voy a nombrar algunas. En el 2016 recibió el Howard Hughes Medical Institute Faculty Scholar Award. En 2009 recibió el Science Age Professor del Departamento de Química de la Universidad de Michigan y el NSF Career Award. Es y ha sido editora de varias revistas importantes en su campo y en campos de biología en general como en eLife, ACS Chemical Biology, RNA, es editora de JBC, The PLOS Genetic, entre otras. Y el día de hoy nos va a hablar sobre Ensuring Ribosome Functionality During Assembly and via Repair. Thank you very much to be here, Katrin, so that we are ready to hear your talk.
Well, thank you so much for this very kind introduction. And even more for the wonderful invitation. I've had a really fabulous day yesterday with the students and today with some of you guys. And so I'm excited to tell you a little bit about the work that we've been doing very recently. So most of what I'll be telling you about is not published yet. So what my lab's really interested in is trying to understand how cells ensure that they have both the correct number and composition of ribosomes. And we know this is important because there's this class of diseases which as a group is referred to as ribosomopathies, and these are linked to defects in ribosome assembly. And what turns out to be the case is that the cells in these patients have both fewer ribosomes, as you can see, as well as ribosomes of different compositions, as I'm trying to show you here with these different colors. And while the diseases display very divergent phenotypes, one common phenotype that all these patients share is a predisposition to very specific forms of cancer, which I'm showing you here. Now, it turns out that the converse is also true. Cancer cells have also dysregulated the number of ribosomes that they have. Most of the time, they have more ribosomes. And more recently, also, it's been shown that the composition of ribosomes in cancer cells is also altered. They lose, the thing that's specifically known is they lose individual ribosomal proteins. And so what this really underlies is the importance of mechanisms that are functional in cells and that my lab has discovered that really ensure the quality control of ribosomes, that ensure that every ribosome is built the same way. Now, how do ribosomes have these effects on protein homeostasis? Well, they do this because, of course, during translation, they have a number of very important functions. Of course, they must first pick the mRNA. And once they then have identified the mRNA, they must also pick the start code on these mRNAs. Now, in addition, of course, they pick each tRNA to ensure a tRNA-mRNA match. And then after every round of peptide bond formation, they must translocate the tRNA mRNA pair through the ribosome. And finally, and this is the most recently discovered function of the ribosome, is that they also detect damage in mRNA in a translation-dependent manner. Now, some of these functions are concentration dependent. Those are the ones that I've indicated here in blue. And this is the reason why both the functional and structural integrity of the ribosome, as well as ribosome numbers, are really important to, again, ensure cellular protein homeostasis. Now, as I said, what I'm going to be telling you about today is mechanisms that are in place both during assembly to ensure that ribosomes are correctly assembled. And then in the second half of my talk, I'm going to be telling you about some other new work in the lab that really has discovered mechanisms that are in play after assembly that, again, are there to ensure that ribosomes are correctly assembled and either lead to their degradation or to their repair. Okay, so to start with the first part of the talk, to quality control during assembly, I need to give you just a 2-second introduction to ribosome assembly. Now, ribosome assembly, of course, starts with the transcription of the ribosomal RNA. And as you can see, an initial transcript is made that contains 3 of the 4 ribosomal RNAs. And this then in the series of ordered events gets cleaved to release the mature RNAs from both the small subunit, which is on the left, and the large subunit on the right. And you can see this is a very hierarchical process, and at least the nucleolar steps are integrated also with the modification of the RNA, the binding of the ribosomal proteins, and the folding of the RNA. And so the outcome of these steps is essentially what most of you guys would really consider assembly. So a largely, not entirely, but a largely matured small ribosomal subunit. And because in my lab, we study really assembly of the small ribosomal subunit, that's all I'm going to be considering for the rest of my talk. Now, while most of assembly starts in the, happens in the nucleolus, these mostly matured molecules are then exported into the cytoplasm, where the final steps of maturation, so specifically the cleavage at the 3' end, so you have to excuse, the pointer isn't working well, so you have to kind of follow my words here. The cleavage at the 3' end and the incorporation of the last 3 ribosomal proteins is integrated with quality control mechanisms. And so so these quality control mechanisms are really functioning, but and we discovered them a number of years ago now and have characterized them. And what happens is that these nascent 40S subunits are binding mature 60S subunits in a mechanism that requires the translation initiation factor EIF5B to make what we call ADS-like ribosomes. And while these ADS-like ribosomes don't have tRNA or mRNA and presumably don't make any polypeptides, maturation continues in them. And when they're fully matured, then they're broken apart by the translation termination factors DOM34 and RLI1 to now release the mature 40S subunit, which then can bind mRNA and start translation. Now, because the cycle looks a lot like translation, we call it a translation-like cycle. And then in a series of work have shown that it doesn't just look like it could be a test drive for nascent ribosomes, but it actually really functions as a way to check out individual functions of the small ribosomal subunit. Okay. And so, as I said, maturation occurs in these ADS-like ribosomes. And of course, the last, one of the last parts of maturation is the formation of the 3' end of 18S RNA. So that happens in these 18S-like ribosomes. And in fact, the chemical cleavage reaction is carried out by this molecule called NOB1. So NOB1 is an RNA endonuclease. It cleaves the RNA to produce the mature 3' end. And in fact, it is helped in this job by its binding partner called PNO1. And so we know that this is one of the final steps in the maturation of the small subunit. And what we had previously shown is that after NOB1 cleaves the RNA, the protein itself, together with its binding partner, dissociate from the ribosome. And now the very last step in 40S ribosome maturation occurs, which is the binding of the final ribosomal protein, RPS26. So these are the last 2 steps in ribosome maturation. And as I already mentioned, they really occur in these ADS-like ribosomes. I just, for simplicity, I'm not going to show them again because otherwise you will not ever be able to make anything out in my cartoons. All right, so a number of years ago, we reconstituted this reaction in an in vitro system that just uses RNA and the protein. And while we were able to show that the RNA, the protein can cleave the RNA at the correct site, which is shown here with this green arrow, you can also see that there are, in this second line, that there are 2 additional cleavage spots. And so what this suggests is that the protein has only limited specificity. And in fact, if you look at the sequence around the cleavage site, this kind of makes sense, right? The sequence is very degenerate. And if you think that the correct cleavage happens between 2 A's, there's a lot of other places where there are 2 A's. And so, so this led us to the question of how is the correct cleavage enforced? And we thought it was really likely to be important that the correct cleavage is enforced because, as you can see here, the RNA, the 18S3' end is completely conserved in organisms from yeast all the way to humans. So if it's not conserved, presumably it's really important that the end is also made at the correct spot. And so to ask if RNAs that are miscleaved are functional, we took advantage of an assay that is kind of outlined here. So as you may know, ribosomal DNA in yeast and actually in all eukaryotic organisms is encoded in many repeats, hundreds of repeats. And so you cannot just make a mutation in the genome because you would have to make literally hundreds of mutations in the genome. And the way that we deal with this is that we use a yeast strain that has a temperature sensitive flavor of RNA polymerase 1. So just by shifting the temperature, we can shut off all transcription from all of those hundreds of repeats. And then transcription of the RNA happens from a plasmid that looks, as I'm showing you here, where that has the entire rDNA operon. And of course, you can then modify this plasmid in any way, shape, or form that you want. You can introduce mutations, or in our case, we made a number of constructs where the cleavage site, where the RNA was shortened by 1, 2, or more nucleotides. And we then asked if these RNAs would be functional. And so we measured growth in quantitative growth measurements. And what I'm showing you here is that if you shorten the RNA just by 1 nucleotide, there's no effect on growth of the yeast cells. But if you remove 2 or 3 or 4 nucleotides, you can now see that there's a defect in the yeast growth. So that suggests that there's something not quite right. Now, the simplest model of what could be wrong is that these ribosomes would be not quite as stable. But what I'm showing you here is that that's not the case. The RNA, the mature RNA, is as stable in all of those mutants as in the other, maybe even more abundant. And we can also show that that these ribosomes are actively translating, because you can also see that the RNA that is specifically coming from the plasmid, and therefore I'm labeling it here with 18S tag because it has a specific tag that we can follow. It's also in the polysomes for all of those mutants, and I'm just showing you 2. So we know that the ribosomes are stable, they can translate, but they are somehow not quite as good at translating, and that's why they have these growth defects. Okay, so what this suggests that yes, it is really, as we had expected, it's really important that the RNA is cleaved at the correct site. And so the next question that we asked is, well, is it then always done at the right site in yeast cells? And so in order to answer this question, we essentially performed RNA 3' end sequencing on on 18S RNA that we purified from yeast. And the first thing that we observed is that really, truly, most of the ribosomes end, you can see, the dots, most of the RNAs end exactly at the site that is indicated with this label D, okay? And that's a very steep step down. But if we zoom in, you can see that while the vast majority of the RNAs is correctly cleaved, there's a small percentage of them that actually end slightly before, and that's shown in the middle. And if we quantify this, you can see that most of the miscleavage occurs at these 2 sites, either 1 nucleotide adjacent or 4 nucleotides upstream. And in both cases, you can see it's upstream of an A, so there seems to be some limited sequence specificity for 5' to an A. And if we look at the miscleavage on the other end, we can also again see that there is some miscleavage on the other end, although it seems to be tenfold less abundant. And we think that we understand the reason for that. All right, so it seems to be fairly rare in vivo, but in vitro, we can see this miscleavage. And it's not just that we see this, you can see there's a whole list of papers from 4 different labs. It's not that we are not good biochemists. The protein really isn't very specific, at least in vitro. And so what it led us to ask then is, okay, what is present in yeast cells that enforces the specificity that is not present in our in vitro reactions? And as we were thinking about this, we actually had some clues, because we had previously shown that this kinase called RIO1 monitors whether or not NOB1 does any cleavage at all. And only if NOB1 does any cleavage does it release the ribosomes into the translating pool. And so specifically, the way that this works is outlined here. So it turns out that NOB1, the presence of the protein itself, blocks the recruitment of mRNA. And so the release of the protein requires both the RNA cleavage, because that makes the NOB1 protein bind less tightly, as well as the ATPase activity of this RIO1 protein, which then releases both PNO1 and NOB1. And so because now NOB1 is gone, mRNA can be recruited, and the proteins can start their first round of translation. And so what we thought then, and in contrast, what we thought then could be the case is that RIO1 would now be ideally positioned to discriminate not just between RNAs that are cleaved, yes or no, but also between RNAs that are correctly or incorrectly cleaved. And so to test this model, we took advantage of a reagent that we had previously described, this mutant in PNO1, which can fall off independent of RIO1. So this is a bypass mutant that doesn't need RIO1, it just falls off by itself. And so if RIO1 really is monitoring this cleavage, then the prediction would be that you get more miscleavage in this mutant. And in fact, you can see this. This is exactly what we see. And this doesn't seem to be random miscleavage, because we can still see the same abundance pattern that we see in the wild type case. And there's not, the difference isn't quite as significant on the other end. Okay. A second prediction from this model that RIO1 is monitoring this cleavage would be that in the miscleaved, because RIO1 releases NOB1 and PNO1, the prediction then would be that in the miscleaved RNAs, when RIO1 cannot function, NOB1 and PNO1 should be retained on the molecules. And so we can't look for PNO1 in this assay, because PNO1 happens to run right at the same molecular weight as, so these are purified over an affinity column as one of the reagents that is coming down too. But you can see that in fact, PNO1 is accumulated on the on these miscleaved ribosomes, which are shown here on the very right, quantified in the gray columns. Okay. So what this really suggests is that RIO1 doesn't just discriminate between uncleaved and cleaved RNAs, but that it also discriminates between correctly and incorrectly cleaved RNAs. And so the next question that we had is how is it doing it? And so what we did is we measured in RNA binding experiments the affinity of RIO1 to either an RNA that ends at the correct sign, which I'm showing you at the top and with the dots, or at an RNA that's either 3 nucleotides too long or 4 nucleotides too short. And what the data shows here is that, in fact, the RIO1 protein binds more strongly to the correctly cleaved RNA than the incorrectly cleaved RNA. And so it is picking out the correct RNA simply by RNA binding activity. Okay, so one prediction of this would be then, because if it is an RNA binding thing, that if you just give it more RIO1, it's an equilibrium, and you can eventually push the equilibrium to the right. So a prediction would be that if you increase the concentration of RIO1, you now discriminate less well against the miscleaved RNAs. And in fact, this is what you can see when we are overexpressing. So here we are now looking at how much of this miscleaved RNA is in the cells. You can see that the miscleaved RNA the -2 and the -4 RNA is more abundant when we overexpress RIO1, which is in the gray columns, than in the absence of overexpressed RIO1. And consistent with this, we also see that these miscleaved RNAs become more toxic because they, there's just more of them now. Okay, so what this really says is that, first of all, that RIO1 helps discriminate between correctly and incorrectly cleaved RNAs. And the second thing that it's really suggesting is that the importance of RIO1 concentration is really critical, because if you have too much RIO1, then you will now allow for miscleaved RNAs in the translating pool. If you don't have enough RIO1, well, then you're not getting short ribosomes into the translating pool. Now, I just want to show you here that we think that this is not unique. We think that RNAs, obviously many RNAs, in fact, probably almost all functional RNAs are transcribed as precursors. And at least in the ribosome assembly pathway, all of the nucleases seem to be kind of imprecise. So this is a nuclease that is involved in the preceding step in RNA maturation. And again, you can see that there are 3 cleavage products. The green one is the correct one. The other ones are incorrect ones. And again, we are not the only lab that sees this. There's one other lab that makes this observation in vitro. And in fact, in vivo, the same observation was previously made. So this RNA cleavage is also imprecise. And the maturation of the 5' end by this other enzyme, UTP1, is similarly imprecise. You can see that it is happening also at an adjacent site. And this is the one that I've shown you. So we think it's quite possible that there's other steps in RNA maturation, and perhaps even globally in RNA maturation, that need to be proofread in similar systems. Okay, so this is just a small story that I wanted to tell you about one of the quality control pathways that are functioning during assembly to make sure that only correctly matured ribosomes are released into the translating pool. But I also wanted to tell you about this because it really leads into the next story that I want to share with you, which is really about quality control after assembly is complete. And so far, everything that I've shown you used yeast cells where either the ribosomes are either all correctly cleaved, like in this on the left, or all ribosomes are miscleaved, like on the right. And so, as I said, all the cells would be either blue or all the cells would be either black. But that's, of course, completely artificial, right? Like cells will never make every single ribosome wrong, right? If they make a mistake, they make a mistake at some small percentage of the time, right? And so what we were asking then is like, well, what happens to the cell when you have a more realistic model, right? Where maybe 1 in 100 ribosomes are incorrect. And so we started to do the same experiments using the exact same plasmids, but now instead of using them in the strain that I was telling you about, where you can shut off the transcription of the RNA polymerase I, we just use them in normal wild type yeast, right? So almost every ribosome is made from the genome, and then there's a small percentage that come from the plasmid. And I should also say that we've also done them in the other strain without changing the temperature, and we are getting the same results. So this is not a strain dependent thing. Okay, and so the first observation that we made is that these ribosomes are now providing us with dominant negative effects. So when we put in those shortened ribosomes, 2, 3, 4 nucleotides shorter ribosomes, they actually grow more slowly than if they didn't have any plasmid in there at all. And we thought this was really surprising because, again, maybe 1% of the ribosomes are now the wrong stuff, right? So this dominant negative effect seems to arise at very low concentration. And it suggested that the wrong ribosomes could globally perturb translation. The second observation that we had made is that even though I'd shown you before that when all ribosomes are miscleaved, these RNAs are equally stable regardless of whether the RNAs are functional or non-functional. When the RNAs are a minority, right? So if they're embedded in a sea of functional ribosomes, now all of a sudden the non-functional ribosomes are no longer stable, right? So that was really surprising. And and this is the case, even though, you know, and they seem to be also depleted from the polysomes. So remember, I told you that the RNAs can translate. We've already shown this when they're by themselves. But when they're now, again, embedded in a sea of functional ribosomes, they are still in the polysomes, but they're now depleted from the polysomes. So when we made these observations, we scratched our heads for a while. And then we remembered a series of observations that has been published in the literature in the last maybe 3-4 years. And this observation is that ribosomes, when they encounter either a damage in the mRNA or just sequences in the mRNA that are more poorly translated, will eventually collide. So this is because the mRNAs cannot traverse that region of damage in the mRNA or the sequence, the special sequence of the mRNA. And so then the ribosome will stall there. The next ribosome will eventually catch up with it. So then you have these collisions. And these collisions are then basically just a platform for the assembly of a whole set of proteins, which eventually disassemble the complexes and lead to the degradation of the mRNA. And so what we were wondering was if essentially in the case where we now have some non-functional ribosomes, we would also see collisions, not because the ribosomes couldn't traverse a specific segment in the mRNA, but because one ribosome was going slower than the following one, and it's like in a traffic jam. Eventually, this one will catch up. Now the ribosomes would move together as in a, like in a tray. Okay. And so to test this model, what we did is we asked if the decay of the RNA required the same kind of molecules that were already described to be important for the disassembly of these collision complexes and the degradation of the RNA. And in fact, you can see that when you delete a subset, so all of these factors are involved in these collisions. And you can see that when we delete a subset of these here on the left, we can see now that the RNA becomes more stable. And for those, then we also see that these miscleaved RNAs become even more toxic. Now, so this is essentially summarized here. So this collision repair process is abbreviated RQC. And you can see that there is a partial overlap between this pathway and the one that we have characterized on the bottom. Turns out there's a second pathway that's been characterized in the literature, and that's referred to as NRD. And this is really for ribosomes that cannot translate at all. So they will be stalled at the start codon because they cannot translate at all. They can make ADS ribosomes, and then they're stuck there. And again, there's a partial overlap in the molecules that are required for this. Now, what this really suggests is that when you have miscleaved ribosomes and they escape into the translating pool, they're then identified by these ribosome collisions in a process that has similarity to this mRNA decay collisions, as well as to the collisions of ribosomes at the start codon, but also has similarity. Now, we're very excited about this. And one of the reasons why we're really excited about this is because I think it has really wide-ranging implications for the how cells monitor the ribosome, the integrity of the ribosome pool. Because of course, it would be a way not just to monitor that ribosomes are correctly assembled, but also if they get damaged over time, or it also has huge implications for this concept of ribosome heterogeneity. Because what it really says is that this is a way that cells will purify the ribosome pool, right? So if you have, what I've shown you is here, if there's one ribosome, the blue ribosome, that is somehow different in a way that it'll be translating slower than other ribosomes. And even if it's just doing this over specific sequences, eventually the next ribosome that is not a blue ribosome will catch up, leading to the collisions. Now, I'm not saying that there's never any ribosome heterogeneity. What I am saying is that I think it's very, very likely restricted to very specific conditions. And one of these conditions could be if the ribosomes sort themselves on different mRNAs. So if the blue ribosomes like different mRNAs than the white ribosomes, then they would be never on the same mRNAs, and therefore they could never collide. And a second condition could be in cells where there's just not a lot of translation. So there's maybe only 1 or 2 ribosomes, right? And if you only have 1 ribosome, then by definition, you can't collide anyone. So if there's not a lot of translation, the distance between ribosomes is long, it'll take a long time for the ribosomes to collide. Now, these are not hypothetical situations. So we already know that in certain cell types, for example, in neurons, there's not a lot of translational activity, and a lot of translation happens actually from ADS ribosomes. And then also during development, it is also well characterized that a lot of translation happens in ADS ribosomes. So I think it's very, if I was looking for specialized ribosomes, those would be places where I think one might want to start looking. And then the other condition I was telling the students about yesterday, it turns out that we've already shown that such a case exists because there are yeast cells make ribosomes lacking a specific ribosomal protein, RPS26, because that is involved in Kozak sequence recognition and therefore identifying specific mRNAs. They sort themselves differentially on mRNAs. Okay. And so this is this work that I was referring to. So it turns out that yeast cells under high salt and high pH stress make these ribosomes lacking RPS26. They recognize differentially different start codon context, which I've shown here, and therefore select different flavors of mRNAs. So these are not hypothetical situations, but as I said, what this really does suggest is that ribosome heterogeneity is not universal. You have to look in niches if you want to find it. All right. So for the rest of my talk, I want to switch gears just a little bit and think about damage to, obviously, to ribosomes, but also to all cellular molecules. And start by reminding you that oxidative stress really does damage all cellular structures. And the cellular structures that I like to think about most, of course, are RNAs and proteins, especially when they're found in ribosomes. As I said, it's already known that oxidative stress damages all of them. And I've also already told you that ribosome collisions in the act of translation is required to identify damage in mRNAs. So again, I want to remind you that this is a concentration dependent process. It's already shown that if you have fewer ribosomes, this doesn't work as well. And so the question that we had then is what happens to ribosomes under oxidative stress? And to start getting at this question, we took advantage of a probe that one of my colleagues, Kate Carroll, developed in her lab. And what this probe does is it labels oxidized cysteines. So it's indiscriminate. It labels every oxidized cysteine. But what we did is we coupled it to and so you can then detect it with an antibody. And when we also coupled it to antibody detection of ribosomal proteins, okay? But I'm already getting ahead of myself. So what I'm showing you here are extracts that we prepared from yeast cells, either without addition of oxidative stress or with oxidative stress, and that we then put over a sucrose gradient to separate the ribosome-bound fractions from ribosome-free fractions. And what I've circled in the red box are the ADS fractions, so it's all ribosome fractions. And you can see that there's a lot of proteins that are lighting up. It's more so in the condition with oxidative stress, but even in the absence of oxidative stress. And these are all essentially in the size range that you would expect for ribosomal proteins. And so to now start identifying individual bands, we used our collection of antibodies against the ribosomal proteins. It's not that big of a collection, but the one protein that really clearly lit up there is RPS26. So I'm indicating here the specific band, and you can see it's one of the most oxidized bands. So we were really excited about this, obviously, because we had done a lot of work on this protein already, and it's quantified here. So what these data say really is that that RPS26 specifically is one of the most vulnerable proteins to oxidative stress in yeast cells. Okay. Now, what would happen to RPS26 when we expose it to oxidative stress or to the cysteine specifically? Turns out that RPS26 has 4 cysteines, and they form a very canonical zinc finger motif that I'm showing you here. And so what we suspected would happen is that if one or more of those cysteines get oxidized, the zinc would just fall out. And to test this, we took advantage of a very well characterized assay in the literature where you use a fluorescent molecule that changes its color upon binding of the zinc that gets released from the protein. And then we made recombinant RPS26. So this is just RPS26, no other ribosomes or ribosomal proteins around. And when we incubate the protein with more and more hydrogen peroxide, you can see that we release more and more zinc. So this suggests that we were right, that the zinc gets released under oxidative stress. And if we make mutants in the protein where one of those ligands is mutated, you can see that they now no longer release any zinc because presumably they don't have any zinc bound. Okay, so what this says is that the zinc oxidation will, the RPS26 oxidation will release the zinc. And what I didn't tell you is that the this zinc finger motif, as you might expect, is actually at the RNA protein interface. So presumably it will release the protein or help release the protein. Now, the next question that we asked is, okay, does this render the protein non-functional? And so to answer this question, what we did is we made mutants in each one of those cysteine residues. And then we asked in yeast, are the yeast still viable? Now, what you can see is that two of those mutants are entirely dead. The third one is essentially dead. And the fourth one seems to have no growth phenotype. And that's apparently very typical for zinc finger motifs, that they really only have to have 3 of the 4 ligands. All right, so to summarize, what this says is that ribosomal proteins get oxidized. One of the proteins most prone to oxidation is RPS26. And when it does get oxidized, it releases the zinc from the protein, and that renders the protein non-functional. Okay, so okay, there we go. It just had a little moment. We all do, right? Okay, so the next question then that we had is, okay, what happens with these ribosomes? And you know, and specifically, we consider 2 models. I just showed you earlier that we had discovered this translation-associated process that can identify non-functional ribosomes and then leads to their degradation. So obviously, it was very possible that this pathway would also lead to the degradation of these damaged ribosomes. But we also thought that this might be perhaps a little bit wasteful, right? Just like if you have a flat tire, you don't trash your car, right? You just get a new tire, right? And so we were wondering if there was a way to essentially just change the ribosomal protein. And one of the reasons why we really considered this model is because we had recently shown that actually there was a way, there's a tool to change the tire essentially. Because we had shown that under conditions of high salt and high pH stress, the RPS26 deficient ribosomes that are made are actually made by specific release of RPS26 from pre-existing ribosomes via a chaperone called TSR2. Now, as I had already alluded to, this leads to our, you know, like these ribosomes that have selective mRNA binding properties. They now translate different proteins that support actually the resistance to high salt and high pH stress. But so what we, and we also knew that this was a reversible process. So what we wondered was if this tool could maybe be used under conditions of oxidative stress, not to make RPS26 deficient ribosome, but to allow for repair of the ribosomes. And so the first question that we asked is, can TSR2 actually release the damaged protein? And the data is here. So you can see that, in fact, under conditions of oxidative stress, so this is a pelleting assay where the ribosomes and everything bound to ribosomes will be in the pellet. Things released from the ribosomes will be in the supernatant. So you can see now in the supernatant is RPS26 when you both expose the cells to oxidative stress and if you have TSR2 there. And we also know that this, the right, I'm showing you that the ability of TSR2 to both bind the 40S ribosome and RPS26 is required for this. So it doesn't just capture released protein, it actually needs to bind to the ribosome to release the protein. So that is summarized here. So the oxidized protein is then can be released, at least in vitro, by TSR2. So we obviously wanted to know, can this happen in yeast cells? And so the first experiment that we did is we asked where is TSR2? And so we did the same experiment where we used those lysates from yeast exposed either with or without hydrogen peroxide. And now we are asking what happens to TSR2. And what I'm showing you here is that it's relocalized from the free fraction in the absence of TSR2 to the ADS fraction, so to the ribosome bound fraction in the presence of TSR2. And that's just quantified on the right. So yes, under oxidative stress, TSR2 starts binding to ribosomes as expected. And then the question that we asked is now using this probe that I'd shown you with the cysteine that allows us to label oxidized RPS26, that the RPS26 actually goes away over time. Now, we don't think that this is degradation of the entire ribosome, A, because it's too fast for this, but also because we were able to show that it actually requires the activity of TSR2. Because if you make these mutants that can't release RPS26 anymore, now the oxidized protein is actually stable in yeast cells. And so the next question that we asked is does the protein where does the protein disappear from? And so what I'm showing you here is that as the protein disappears from the ribosome bound fraction, so that's on the left, it first shows up in the free fraction. And what this really suggests is that, again, it's consistent with this model that TSR2 releases the protein, and then the released protein is what's presumably getting degraded. And this now leaves, of course, a ribosome lacking RPS26. And so this now looks like almost like a ribosome as it's being made, because remember, the last step in ribosome assembly is actually the incorporation of RPS26. And so we wondered if now RPS26 could be reincorporated, and it's already known that that's the job of TSR2. And so to ask if TSR2 can do this, we first again used an in vitro system where we are first, on the left, we're first releasing RPS26 under oxidative stress, then we're purifying the ribosome, and we are then adding recombinant TSR2 and RPS26. So when we do this with wild type RPS26, we can see that the RPS26 gets incorporated into the pellets, so into the ribosomes. But it turns out that actually TSR2 can discriminate against, or the TSR2 and the ribosomes can discriminate against correctly and incorrectly folded RPS26, because with the mutant RPS26 that don't have the zinc, they do not actually get incorporated. And so the last experiment that we did is to confirm that there really is a repair is this pulse chase experiment. That is a variation of an experiment that I already showed the student yesterday. So here we asked if RPS26 gets extracted and then repaired, and the ribosomes get repaired with new RPS26, then you should be able to find new RPS26 in old ribosomes, specifically under oxidative stress. And to test this, we took advantage of this yeast strain where we can label old ribosomes with an affinity purification tag on RPS3. So that's the green little tag there, I guess. And that's inducible under a Gal promoter. So we can turn it off. And so that way we know that all the ribosomes were made before we turned it off. They are old ribosomes. And then the new RPS26 is under a different promoter. In this case, we used a TET promoter, and it's labeled specifically with an HA tag. Okay. And so when you look here, and so we've now, I'm just showing you the purified ribosomes, the old ribosomes that were affinity purified using the tag. And you can see here that, in fact, the HA tag new RPS26 shows up actually in ribosomes, even without oxidative stress, but much, much more so when you expose the yeast to oxidative stress. So what I think this really does is it proves that old ribosomes can be repaired with new RPS26. This seems to be happening constitutively in yeast, and that's not surprising because there's always oxidative stress. But when you add hydrogen peroxide, this is clearly hypercharged. Okay, so this is what I've shown you so far, that under oxidative stress, RPS26 gets oxidized, TSR2 releases the protein, and this then produces a substrate for repair with newly made RPS26. Now, does this matter? Yeah, we think it does matter, because when we mutate again TSR2 to make it non-functional, these yeast cells are now slightly more sensitive to oxidative stress. Now, I just want to remind you, we have now just change the repair of one single protein. There's 79 protein in the ribosomes. There's, 6,000 proteins in yeast, right? So just messing with one protein makes these yeasts more sensitive. And then the second question that you might ask is does this only happen to RPS26? And so the answer is no. So when we looked at both the proteomics data for yeast and the number of different organisms, we noticed that there were two proteins that were oxidized in all cells that were analyzed, in all organisms that were analyzed. One of them is RPS26. The other protein is RPL10. And actually, when we analyzed our mass spec data for which residues on the ribosomes are oxidized, RPL10 also was in there. And so turns out that RPL10 is another one of the ribosomal proteins that have their dedicated chaperones. And so obviously we asked the question, can this also be repaired? And so the answer is yes, otherwise I wouldn't be talking about this. So what I'm showing you here is if you add the chaperone for RPL10, which is called SQT1, and you expose the purified yeast ribosomes to oxidative stress, then also a fraction of the RPL10 can be released in the supernatant. Just like I've shown you for TSR2, SQT1 gets recruited into the 60s and 80s fraction when we expose the yeast to oxidative stress. And just like for TSR2, the ability of SQT1 to interact with the ribosomes and with RPL10 is required for the disappearance of the oxidized RPS. And finally, also, this is required for the oxidative stress resistance because, again, if we disable this machinery for one other ribosomal protein, again, you render the yeast more sensitive to oxidative stress. So we think that this might be a more general pathway. It's for sure not functional for all ribosomal proteins, but for a subset of ribosomal proteins, what I've shown you is that when they're damaged by oxidative stress, they can then be released in a manner that's specific to each ribosomal protein by an individual chaperone, which then allows them to be repaired with a newly made copy of the same ribosomal protein. So in a way, it's a way to change your tire and not needing to buy a new car. Now, why do I think this? Why do I keep making this analogy? Why do I think this is important? So the first pathway that I've shown you, this collision-induced degradation pathway, right? It obviously requires translation. And one of the outcomes that I've shown you is that it lowers the ribosome levels, right? And because it lowers the ribosome levels, it could then impact the detection of damage in the mRNAs via this pathway that's called RQC in the literature. Now, could this be potentially a really bad thing during oxidative stress? Well, why could repair be more, ribosome repair be more useful under specifically conditions of oxidative stress? Well, first of all, it doesn't require translation, and oxidative stress completely downregulates translation. But probably more importantly is it conserves ribosomes. And so oxidative stress, like many other stresses, blocks the production of new ribosomes. And because I've shown you that actually active translation and therefore ribosome concentrations are really important for the detection of damage in mRNA, which is also ubiquitous under oxidative stress. Repairing the ribosomes will actually likely enable the mRNA quality control under conditions of oxidative stress. And if you just degraded them, then you would take away the ability to detect and damaged mRNAs. So just to summarize, I've shown you one short story, really, of how during assembly proofreading is used to ensure that only correctly matured ribosomes are going into the translating pool and not incorrectly ribosomed. And I've also shown you essentially a mop-up pathway that functions after assembly to identify either misassembled ribosomes or likely also ribosomes that might otherwise get damaged over time and leads to their decay in a translation-dependent process that requires translation and occurs inside the open reading frame. And finally, I've shown you that also under some conditions, at least under oxidative stress, a subset of the ribosomal proteins can be repaired when they're damaged. So lastly, the most important thing, of course, I need is to thank the people in my lab who did the work. So the work on Rio 1 was done by a graduate student who recently graduated, Melissa Parker, and who's now working in a biotech company in the San Diego area. And then Jason Yang is a really fabulous postdoc in my lab who has done all of the work on TSR2 and RPS26 and discovered both this release and repair pathway. And then finally, I want to thank you for inviting me and for you guys all for showing up, and hopefully you have some more great questions from the students.
Thank you very much, Katrin. Very nice and interesting talk. So now the seminar is open for questions.
This pathway also conserved on the mitochondrial ribosome.
So we don't know. So you know, so I don't know that it's known. So the collisions, it's not even known whether those are conserved, but I think it's likely that they could be. And the reason why I'm saying this is that bacteria do the same thing. There's also collision-mediated pathways in bacteria, although they need a protein mut-S, which I'm not sure if that's imported into the mitochondria actually, or something that looks like it, right? Like it doesn't maybe have to be the same protein. But I also don't know. The other thing that you need for this is obviously, because, you know, as I indicated, a high enough translational load. I'm not so sure how high the translational load in mitochondria actually is. So the literature doesn't say anything about it. I'm not so sure about the repair. I don't know. So those are cytoplasmic proteins. We don't, I mean, not to any of, not to my knowledge, are they in the mitochondria? Not also, so RPS26 is a eukaryote specific protein, but RPL10 is not actually. So, I mean, I think it's possible. Mm-hmm.
Hi, thanks for the super nice presentation. I also was thinking about, because I mean, in our lab, we also work with ribosomes, and when we arrest the ribosomes, we see shorter versions of the proteins, and we call this like a ribosomal stacking. So have you ever tried to do that, like use different isolated lysates and then combine them from the blue ribosomes and the gray ribosomes and check whether you start like shorter bands start appearing? So this is part of this process that I didn't talk about. It's actually quite well characterized. So when the ribosomes collide, as I said, this entire machinery then assembles on the collided, at the interface of the collided ribosomes. And so one of the outcomes is that the mRNA gets degraded, and that's the one that matters more for me. That's why I like to think about it. But it is another outcome, obviously, because this is already a translating ribosome. So you also need to take care of the peptide, right? So there's a second branch of this that will essentially unstick the peptide by putting on cattails, so CATs, cysteine, alanine, threonine tails. And so they kind of just pushes, they do this until the protein is pushed out of the exciton channel. So you will like this, right? And then there's a ubiquitinase that then ubiquitinates the protein, and the whole thing gets degraded that way. So it's actually fairly well characterized. I'm not sure that anyone has done the specific experiment that you are suggesting, but you know, but as I said, it's fairly well characterized both in bacteria and in eukaryotic cells and in human cells. And the pathways are similar, kind of homologous proteins. So,
yeah.
Okay.
Yeah. The specific molecules are different, but the way that it functions is exactly the same. Yeah.
And I have another one related with which kind of tires can be changed, because maybe do you think it's related with how essential is the protein, or how, because some proteins have this very long coils that go really deep into the ribosome. Do you think this could also be changed? Or?
I don't think so. And I mean, so the short answer is there's about 12 ribosomal proteins that have these chaperones. Most of them, except for one, are really located on the outside. And I think that makes them both candidates for doing this, right? Because I mean, I was telling the students this analogy that I like to use yesterday of Legos, right? A ribosome is like a Lego project, right? You have a lot of pieces that you have to put together in the right way. But just like with the Lego piece, right? It's easy to take out a piece on the top and replace it, but you can't take the one on the bottom without disassembling the whole thing. So I think that's the question that you're asking. So but the proteins that have these chaperones are all located on the outside. And I think this makes it both possible that they can be replaced, but I'm guessing it also makes them candidates to be susceptible to oxidative stress. Because again, if they're on the inside, then it's much less likely, that they will become oxidized. And so I've shown you 2 proteins that get repaired. We have fairly good evidence that there's at least a third. And then I told you we have basically analyzed the proteomics data and oxidized proteomics data for a number of other organisms. And I'd say there's at least one other good candidate that we just haven't been able to test because there's no good antibody against it. And even in vitro, so it's hard to, and in vitro, the band overlaps with some, anyways, it's weird for stupid reasons we haven't been able to do it, essentially, yeah.
Thank you.
There's another question.
Thank you for your nice talk. My question is regarding oxidative stress in cancer cells. Do they have increased this repair mechanism? And whether that correlates with more aggressive stages or not?
Yes, so you can, yes, that seems so. TSR2 is upregulated. There are cancer cells where it's highly upregulated, and there are also others where it's downregulated. So I think it's complicated because it is also required for incorporating RPS26 in the first place. So I've kind of hidden this in the experiments. So when you have no TSR1, I'm sorry, TSR2, that's a different protein, TSR2, or you have these mutants, actually the yeast cells have more ribosomes lacking RPS26. So we have to fix this by overexpressing RPS26 in the background. If we do this, then we can fix this. So and then it's already known that having ribosomes lacking RPS26 this is one of these diseases that lead to cancer predisposition. So it's very complicated. So not having enough TSR2, I think, produces cancer because now you have the ribosomes lacking RPS26. If you have too much, that's also associated with cancer because now I think, I think maybe you get release. That could be some of it. And then the repair, yeah, that's another whole question. So, but yes, it's hard to figure out. There's definitely changes in TSR2 that are associated with cancer, but to fully understand what they mean is challenging because of that.
Thank you.
Mm-hmm.
So I don't see. Oh, there's one question in the chat, but the question is: are these analyses made in vivo or in vitro?
So that's a good question. So it should have been probably more clear. So we've shown this. So all the stuff for the repair was done both in vitro as well as in vitro, so in vivo. So we can show that all you really need is the ribosomes, purified ribosomes, recombinant protein, and it can release the protein, and then it can also reincorporate the protein. And we've also shown that this happens in vivo. And I think the strongest experiment there is really the incorporation of the new protein into the old ribosomes. And the stuff about the collision-mediated decay, that was all in vivo. There was no, other than this one RNA binding experiment.
Okay.
Yeah.
So there is one question in the chat. Marta Vasquez raised her hand. Do you want to perform your question, Marta, or?
Yes. Hi, thank you for your talk. Do you know something about simulation of ribosomes under stress conditions?
Simulation of ribosomes under stress conditions.
Simulation? So we haven't looked at simulation. We have been asking what happens to the protein when it gets released. How does it get degraded? And so there we're testing the specific model that it is ubiquitin mediated decay. And this is not just because it's the most obvious, right? But because it's also already known that actually ribosomal proteins are produced in excess, and the ones that don't get incorporated into ribosomes during the production of ribosomes are then degraded in a ubiquitin-mediated pathway. So it's already precedent that ribosomes that are just floating around, ribosomal proteins that are floating around outside of ribosomes get degraded in a ubiquitin. So now we're testing if this is also required for this decay, but we haven't looked for sumorylation. Yeah.
Thank you.
Mhm.
Do you have a question, Christian? Yeah, go ahead.
Well, thank you for the talk. I was wondering, I really like this collision-induced degradation, but for it to work, there must be a certain distance that is allowed between the ribosomes. Now, is it known how this is or are there mutants known that they're deregulated in this way, or is it known how this works? Because if they're getting too close, then they should collide all the time, no?
Yes, so it's actually, I think we're starting to fully understand it. So the distance is known to matter, and the probably best piece of data for this is when you change ribosome concentration, you limit the collisions because now you increase the distance, right? And I think, I'm pretty sure that paper also had something about translation initiation, because that is, again, that is the other thing that limits it, like for well-trained. And so there is evidence there, also from reporter assays that better translated mRNAs are more prone to these collisions and decay. So I think there's good evidence for that. And so what limits the distance shortly is this frequency of essentially loading, and then you had a second question that was really interesting. There was a follow-up of this.
If there are mutants, maybe, or deregulated, if there's some.
Okay, so does this affect some things more than others, is essentially you're asking. And, oh, no, this should happen all the time. Now I remember. So it seems like that there are, I think, multiple outcomes from this, and they depend. I think it's not super well characterized, but what I think seems to happen is that if you have just a few collisions, then there's one protein that binds these interfaces, and it essentially does a bunch of different things. And one of the things that it does is it downregulates new translation initiation and globally. And so that should limit all subsequent collisions if this is just kind of something transiently what happens. If this is not enough, then I think that protein is limiting. And so if there's more collisions, then that protein is abundant. Then what seems to happen is that then a different set of proteins can get recruited to that interface. And these will have the more drastic outcomes. And so there's essentially in yeast cells, there's two outcomes. In human cells, there seems to be a third outcome that eventually actually leads to an apoptotic cascade. So I think the amount, so I think this is a way to essentially measure the frequency of collisions. If you just have a few, well, you just globally downregulate translation, hope everything goes away and is better. If there are more, then you're starting to take more drastic measures. So I think it's still a very active area of research, I think, from a lot of labs that are not mine. Yeah.
Can I ask another one, a final one? I just remembered in the first part, you talked a lot about this final processing of the RNA.
Mm-hmm.
Is there a reason known why this is the final step? Because I seem to remember from, let's say, biochemistry or molecular biology that there's a kind of lots of processing going on of the rRNAs. So it's a kind of endonucleases, and then there's some exonucleases doing a fine trimming, and there are even, I don't know, modifications of the bases. But why is, well, I would expect that first make all the final pieces of RNA and then set together the Lego. But this seems to be kind of odd to end with this.
I guess, I mean, I haven't thought about this. I don't know that I think you want to end, because the proteins also protect the RNA, right? I think it would be probably challenging to first make an RNA and then assemble the proteins, because you would just constantly get degradation of the RNA. So I think that's probably one practical reason. Why exactly this is the last step and not something else? I mean, I don't know. I think it could have to do with the fact that, well, one thing that maybe, I mean, this is obviously, there's no way of testing this, right? We're just blindly speculating here. So remember, the second to last thing is the maturation of the RNA. The last thing is the incorporation of this protein RPS26. And what I've been kind of telling you in passing is that RPS26 is also involved in the recognition of the mRNA. So, I think it is not hard. And then I told you that the NOB1 protein, the nuclease, is blocking mRNA recruitment. And so these things are all sitting where the mRNA will be. So if you want to, I think it's also therefore just technically easy to make sure that you don't get mRNA until this stuff is gone. So to release it very late, I think, therefore makes sense. And then, yeah, I think so that could be a practical other reason. Mm-hmm.
Thank you very much.
Mm-hmm.
I have two questions. So the first one is, there are some transcripts that are either bicistronic or multicistronic.
Mm-hmm.
So I wonder what happened with translation of these bicistronic RNAs, messenger RNAs, when you have an heterogeneous population of ribosomes. So the collision affects more the translation of these bicistronic RNAs, or it doesn't matter?
Well, I mean, these are super, super rare in eukaryotes.
Yeah.
And it is, I think the reason why is that people think that they are degraded through the nonsense mediated decay pathway, because they look essentially like nonsense transcripts, right? Because they have the stop of the first open reading frame would be really indistinguishable from a premature stop on the second open reading frame. Do you see what I'm trying to say?
Mm-hmm.
So it is believed that is what's making them one of the reasons why they can't exist. But that being said, we know that they exist, right? Like, I mean, everyone now knows how the COVID genome is made, right? And that is actually the exact same thing, right? The proteins, these mRNAs, are polycistronic, right? And the last protein is translated from an mRNA that has whatever I can't remember how many of them, 8 or something, a large number. So how the virus suppresses the NMD pathway there is not known. And with the virus, that's one of the cool things about viruses. They always tell us something about normal cellular function. So if, the short answer is it's probably super rare and there's no clue about.
In Drosophila, there are about 200 transcripts that are bicistronic.
Interesting.
And the proteins are functional. So actually, one of the components of TF2H is encoded in a bicistronic transcript.
Very interesting. So see, that's what I'm talking about. So very interestingly, so if this then involves suppression of the nonsense mediated pathway, that's why I've been kind of thinking about this, and I just haven't been able to figure out how it could work, and therefore an experiment to address it. So RPS26 actually binds directly to UPF1, the basically initiator to the entire NMD pathway. We also know that we have some unpublished data that RPS26 is important in translation termination, as it makes sense, right? If it binds the mRNA during start codon recognition, unless the mRNA changes the path through the ribosome, it'll still recognize the stop codon also. There's some evidence that that's true. So I have oftentimes wondered if that could be a way to if there could be a way to suppress initiation, NMD on transcripts where you want to suppress it, but I don't have any data for that. It's complete speculation. And we also picked it up. We did once we did a genetic screen. So I was telling this is one of the things I was telling the students yesterday. When the NOB1, a non-functional NOB1 is dominant negative. And so we did a screen to look for suppressors of that dominant negative. And so that's how we figured out 301. But we also did a deletion screen using just a bunch of yeast strains that are defective in some RNA decay pathway or the other. And UPF1 actually showed up in that, and the subset of the NMD pathway, not the entire NMD pathway. And so I think it has to do somehow, but as I said, I haven't come up with an intelligent model that's good enough to waste, like to put, someone needs to work on this. And if they're working on this, they're not working on something else, right?
Thanks. So my second question is, I'm curious about this plasmid that you introduced with the complete ribosomal RNA genes, the complete ribosomal DNA that it is transcribed by RNA polymerase II. So, and I wonder if this plasmid or this episome forms a nucleolus, and how many copies you require to, if it happens, it forms a nucleolus, how many copies require just to form the nucleolus using this episome that express the ribosomal DNA.
I can't remember if it makes a nucleolus. I think it does. I know the question has been answered, but I just don't remember the answer. The copies, it's fair. I mean, it's not one. I don't know the exact number, but it is a gal. Is it a 2-micron plasmid? Sometimes, in most cases, it's on a 2-micron plasmid. So that means it's maybe around 10-ish or so. And it's under a gal-driven promoter. So it's like the strongest promoter, essentially, that yeast has. So it's probably, the abundance is probably about, somewhere between 1 and 10% of normal ribosomes.
But it works. It works.
But it doesn't work well. I mean, the yeast grows slow. There's a reason why there were 400 million data points there, because the data is so noisy and the students hate that experiment with a passion. It's a really crappy system, but it's the only system that we have. And so, yeah.
Okay.
Yeah.
Well, thank you very much. I think we have enough questions. Well, fantastic talk. I mean, I always enjoy this pure molecular biology. Oh, there's another one. Okay, go ahead.
Yay.
I was just gonna ask should we expect to see a downregulation in these quality control systems in cell death, like apoptosis or necrosis?
That's interesting. I mean, I think it is possible for sure that you could get the short answer is, again, I don't know. I haven't done the experiment. The molecules are conserved from yeast to humans, right? Both TSR2 and SQT1 have human homologs, and that's true for many of the chaperones. So, I think it's possible that this could happen, because if I go back just one slide, right? Presumably, although we also haven't shown this, but what I'm guessing, well, okay. So one possibility is if you disable the repair, right? Because mess up with those chaperones in the way that we did. One possible outcome could be that now you're getting more of the decay that is induced by collisions, right? We've tried to look for this a little bit. It's not super easy. And so if that is true, and then, I remember the answer that I gave to the previous question, right? If you have too many collisions, ultimately it is known that you get apoptosis, at least in human cells. Yeast obviously can't do apoptosis. So I think it's possible. But I guess because we don't work in human, well, we sometimes do, but we haven't done this in human cells. We haven't looked for it, but that's probably actually a good experiment. And to look for the intersection between the two pathways, that's definitely possible. And I want to look at that a little bit more.
Thanks.
That's a great question. Nice job.
Okay, great. So now we finish. Okay, perfect. Thank you very much. So we appreciate it. And, thank you to all of you. Gracias.